What's today? I don't know what day it is. Is that good? Is that bad? Is that good? Okay, so the main, did we finish this slide?
Yes.
Okay, so the main thing here is that this is the main mechanism where these guys get gases into the gas bladder. And we talked a little bit about why that happens. So I really want you to make sure you understand this process. So, you know, the main point here is that you have a pigment that's carrying oxygen through the blood vessels towards the gas gland. That pigment is hemoglobin, and it holds on to oxygen tightly, unless what? Unless pH is low. And that's the job of the gas gland, to secrete material into the capillaries that are associated with that system, so that pH drops. That changes the affinity of hemoglobin for oxygen. Oxygen is released into the bloodstream or the plasma, and then that increases the concentration. The other important part of it is that the capillaries are arranged in the countercurrent system. So as this blood vessel or this capillary is running alongside this capillary in opposite direction, because of the transition of the pH along this length and what happens with the affinity for hemoglobin, no matter what the concentration of oxygen is here, it's higher than what it is here. And that just kind of allows the diffusion of oxygen, a movement of oxygen from here to there, and again, making it exposed to being able to be pushed into the gas bladder. So two main things, pH and hemoglobin, and then the countercurrent exchange system. That's how fishes get gases into the gas bladder. To get gases out of the gas bladder, what do?
They burn.
That's one thing they can do, but only if they're what kind of fish? What is it? Physostomous, because now there's a connection of the gas bladder to the digestive system, you can let it go. If they're not that, which most fish aren't, what do they have to do?
Just at the ovale.
Right, so the muscles around the ovale, the radial muscles pull, that exposes that membrane, which is permeable to oxygen, to the capillaries, and oxygen that now can diffuse from the gas bladder into the gas bladder, or into the bloodstream. That decreases the volume of the gas bladder, and the fish then descends in the water column. Okay, so that's essentially how it works. I should probably plug this thing in here. Otherwise, my pointer is not talking to the computer. I went to, well, you know, you guys know the bird conference was at the Discovery Park, and they have the TV screens and these pointers. Everybody was trying to point with these things, and TV screens just suck it right up, and there's no pointer. But we keep trying. Point of view, nothing's happening. Okay, so the other thing I wanted to kind of talk about is just the advantages, I guess, and the vulnerabilities that come from having a gas bladder and being able to use it. So the advantages, the main advantage of a gas bladder is that it allows animals to adjust the volume of that so they can increase or decrease their buoyancy. They can move up and down in the water column or hover in one spot without using energy, but also without moving, right? And so a lot of animals, that's going to be important. Why would it be important to not be able to move, or to be able to not move? If you're hovering in the water column, why would you want to not be moving around all the time? What advantage could that bring?
You don't use as much energy.
You don't use as much energy, but what else? Right? So energetically, it's less expensive to just hover instead of swimming. But what else may a fish be vulnerable to or have access to if they're motionless?
They can wait for prey.
When they're hiding, when they're waiting for prey to swim by. So if I'm constantly swimming around, then I'm making my presence known to all the other animals that have really fine sense of vibrations in the water column. You're going to give yourself away, and you're not going to get to eat. On the other hand, if you're on the other end of that relationship, if you're a prey item, you can do the same thing, but now you're avoiding predators finding you because of all the vibrations you're making. So the main advantage, of course, is energetics, but also it helps these animals remain still because that's part of their ecology in terms of finding prey or hiding from predators. And here's a barracuda just hovering there in the water column without moving. And then here's one that doesn't have a gas bladder. I don't know why that's an advantage for that guy. So that guy doesn't have a gas bladder. That's a darter. So there are some species that don't have gas bladders. This is, of course, is a darter, which are benthic animals. And of course, in this case, the advantage of not having a gas bladder is it decreases the buoyancy, so it helps these animals sit on the bottom. And not only are they sitting on the bottom, or they're benthic, but they're usually in moving streams, right? So being on the bottom hard is a good option for these guys, not just kind of resting on the bottom because the current is going to push them around if they have a space inside them that's filled with air. Some animals can use the gas bladder as a source of oxygen storage or oxygen acquisition. We've talked about some of those guys, especially the ones that are physostomous. Many of them have divided gas bladders that have more blood vessels associated with them, and more places where oxygen can diffuse from the gas bladder into the bloodstream. Another thing that the gas bladder provides in terms of just its function is increased hearing abilities. And so what kind of fishes have that? Two examples of fishes that have increased hearing abilities because of the gas bladder. There's two conditions that we talked about that relate the gas bladder to hearing. This was on your exam. Y'all know all this already. You just have to barf it out.
The shadows and herrings.
Those animals that have the otophysic gas bladder, ones with the anterior projections into the inner ear, so the gas bladder is now part of the inner ear system, and the vibrations that are received in this giant bag of air are now transferred to the inner ear. What else? Who else? Go ahead, Ron.
The ones with the Weberian.
The ones with the Weberian, all the Saluiformes, all those, the Asteriophysi, those animals, catfish, all those guys have Weberian apparatuses that basically do the same thing, except they're linked together with bony elements or ossicles. And so not all animals connect the gas bladder to hearing, but some of them do, and the ones that can have an increased ability to do that. Okay, so disadvantages of having a gas bladder relate to the fact that the gas bladder is much less dense than the surrounding tissues. Generally speaking, the tissues of fishes and the density of fishes are pretty close to the density of the surrounding water column. So as vibrations move through the tissue or move through water, they move through the tissue about the same frequency and amplitude, unless they hit something that's not that, right? And so one of the things that's not that would be the gas bladder, much less dense than the surrounding water column. And so some organisms can use that as a target. I'll go back to this one in just a minute. And that would be the animals that have some type of acoustical signaling that can send out vibrations essentially and then respond to what comes back. If you've got this big body of air in the middle of a dense fish, then that vibration, that bounce back is going to be a little bit different than the rest of the fish. And so things like dolphin can find animals in this big water column. Everybody has the same density except for the gas bladder, and that's going to be a problem for animals that are swimming around that are vulnerable to predators like that. The other thing that is not typically a problem, but under some circumstances, having a gas bladder, it doesn't necessarily decrease the range of depth that you can occupy, but how quickly you can do it, right? So with the idea being that if we're talking about a fish that's 30 meters below the surface, there's a lot of water pressure on that animal, and that's going to affect the gas bladder. As they slowly increase the volume of gases in the gas bladder, they'll move up a little bit. But as they move up, what happens to the gas bladder? It gets bigger on its own just because of the change in density. So now you have to be able to really finally control that. So now you have to get some of the gases out of the gas bladder to optimize that particular depth range in terms of your buoyancy. Same thing that a diver does with their buoyancy control vest. If you're on the bottom, you put a little air in your BC and it comes up a little bit, and now as you come up, it's expanding volume, so now you have to let a little bit out so that you can match the density. If you don't, what happens?
You keep going up.
not only do you keep going up, but what's your rate? Say it. Is it, the rate increases. You're going up faster and faster because now you're expanding quicker and quicker, and it's expanding. And so what's happening with fishes, if they go up quickly, then they're going to, that gas bladder expands faster than they can decrease the air in the volume, and then in the volume of the gas bladder. And so what that tends to do then is cause some type of injury, right? Because there's other stuff in the body cavity other than the gas bladder, like what? Huh?
Intestines.
Digestive tract, liver, all those things, everything that's in there is going to now be squished. And so, if you've ever been deep sea fishing or ocean fishing with the the electric reels, you hook a fish and you bring them up, and then you get them up and their guts are coming out their anus because the gas bladder is taking up the entire body cavity. There's no room for anything else, and so obviously that's going to be a problem. That's artificial. But it still limits how quickly they can't go from the bottom and shoot all the way up to the top to catch a prey item because of that. So it limits their ability to change in depth rapidly over large distances. Okay, does anybody have any questions about gas bladder? Sweet. In that case, I'll end this show. We'll start a new show. It's going to be called something. Urogenital system. Let's let me call that. Want to? Okay. Okay, so the next topic, this is all, again, internal anatomy, physiology type stuff. So the next topic is the urogenital system. And so that's upside down. That's a combination of the urinary system and the reproductive system. Okay, so what I'm going to do today or into Thursday if it takes this long, is I'll talk a little bit about the urinary system, what the main function is, and then we'll talk about the reproductive systems in terms of how they're different among the major groups of fishes. So I'm not going to get into great detail of any of these systems, but then I'm going to come back to the urinary system later when we talk about homeostasis and maintaining equilibrium with respect to water and solutes in the bloodstream. So I'm going to talk basically about how it functions, and then we'll come back to it and talk a little bit about some other mechanisms that fishes have for maintaining homeostasis. That's a different topic. Okay, so what we got here? Okay, so and the main reason why I'm putting them together, for now at least, is because for most fishes, most of them, there's a fairly close association between the urinary system, which involves, what's the main structure or the main organ that's involved in the urinary system? The kidneys, right? So there's kidneys, and then of course the testes and ovaries. For males, particularly of fishes, the testes in some groups are really closely tied to the kidneys. So the urogenital system is kind of a combination, and some of them have a common location where the materials are emptied. And so typically in fishes, there's a pretty close association between these two things. Okay, so the main function, of course, is to transport nitrogenous waste, and so that's the role of the kidneys as the organ to get rid of nitrogen compounds. And so that comes from the digestion mostly of proteins. So as animals digest proteins, we talked a little bit about that. A protein is a long chain of amino acids. When a barracuda eats another fish, he's eating those other fish proteins. This gets broken down into the individual amino acids that make that up, and then ultimately in the cells we'll rearrange that to make a barracuda protein. But this process produces nitrogen-containing material that is toxic. And so we can't not do this. And as a consequence of doing that, you'll always have some nitrogen-producing material, mostly ammonia, right? That gets produced as a consequence of digestion of proteins. We have to get rid of it because it's toxic as it builds up. And so that's what's ultimately one of the things that happens in the kidneys is nitrogen-containing compounds, ammonia or urea, is is being removed from the bloodstream so that it can be excreted from the organism. Okay. And so different types of organisms use different products. We'll talk, most fish are going to excrete their nitrogen-containing compounds as ammonia, but some convert it to urea first because that's less toxic, and so there's other things associated with the relationship between water balance and stuff. So we'll talk about those things later, but that's essentially what your kidneys are doing. You're providing a place where ammonia or urea or whatever it is can be removed from the bloodstream and gotten rid of. The units that do that are mostly the glomerulus, and then the nephron. So these two things work together to accomplish what I just said, to get rid of ammonia and other nitrogen-containing compounds so that it can get released from the organism. The other part of it, of course, is to produce and transport the gametes. And so if we're talking about males, the male organ is the testes, and that's where meiosis occurs and producing sperm cells. And then the ovaries is where meiosis occurs to produce ova. And then, of course, transported either out of the organism or, so there's some variability in terms of how the gametes get together in fishes, but ultimately that's going to be the same. And some of them have a stronger connection with the kidneys. And so that's why we're talking about these things together. Okay, so in sharks and the sarcopterygian fishes, who's the sarcopterygians? When I say that, who do I mean? The lungfish, lobe-fin fish, that group. And so in sharks particularly, and some of these guys, the materials are transported along the same paths. So some of the ducts and channels that are part of the urogenital system are the same for some of these animals. And as you get to the more derived groups of fishes, they become more separated. And so that's going to be kind of the overall discussion for today. And so for these guys, for example, the materials from the urinary system, from the reproductive system, empty into the cloaca, which we talked about before. That's what makes it a cloaca, because it's receiving all this material from different places, including the digestive system, and then ultimately is released from the organism from the cloaca. Other organisms, those things are separated. You've got the digestive system, the anal opening, and then you've got the urogenital pore, where the urinary material and the reproductive material. Right, so in most of the more derived species, those are going to be transported in separate tubes or ducts and eliminated from the organism or released from the organism through the urogenital pore, which is a pore that releases what? If you're a male, for example, the urogenital pore is releasing what? Sperm is one thing. What else? What's the uro part?
Urine.
Well, could be urine, but in fishes, not really urine. One of two things. Ammonia or urea, the two things that fishes release. So urine is a form of nitrogenous waste that is particular for certain types of organisms. Okay, so yeah, that's what's happening at the urogenital pore. Or if in some, like you've got two separate, you've got the urinary pore and then you've got the genital pore, and that's more common for females to have a complete separation of the exit points of the materials. Okay, so a little bit about the structure of the kidneys, just so you see how it works, and again, we'll revisit this later, so I'm not going to get into great detail here, but so the main function of the kidney is to filter the blood, so that, filter is not really a great term, but expose the blood to different permeabilities so that nitrogenous waste is removed. Before I go any further, this is the main function for terrestrial organisms, for most terrestrial organisms. Fishes less, although this happens in the kidneys in fishes, but fishes don't really need to use the kidney as much, most fishes don't, to use the kidney as much for ridding ammonia. Anybody guess why that would be true, as opposed to a terrestrial organism, for example?
Would it be less diluted or more diluted in fish since it's in water?
I mean, not in the surrounding environment, but you're on the right track. There's something about living in water that's different, right, than living on land. And so, maybe I'm asking too much here, but ammonia in its pure form is very toxic. So you really have two choices. You can get rid of it really quickly or turn it into something less toxic. And that's what terrestrial organisms do. Mammals mostly turn it into urea. Reptiles mostly turn it into uric acid, which is much less toxic than ammonia. Fish don't. Fish, ammonia is what they mostly get rid of their nitrogenous waste. That's what the main form is. Why is it that fish can do that we can't? You're kind of on the right track, I think. They can get rid of it a lot more quickly. How come? Okay, let me approach it like this. For us to get rid of it using our kidneys, it has to be where first? How does it get to the kidneys? From the bloodstream. So the bloodstream is delivering it to the kidneys so that the kidneys can filter it out. Fish don't have to send it to the kidneys, freshwater fish particularly, because they can get rid of it in another spot much easier. So it has to be a place where there's a rich supply of blood, right? Because that's where all the nitrogenous waste is happening. So is there a spot where a fish could get rid of ammonia quickly because there's so much blood passing through it?
The gills?
The gills, right? So it's not just because it's the gills, but because the gills are also exposed to what? Water. And of course, ammonia is soluble in water, and it just diffuses right out of the gill membranes because it's being flushed with water that's very low in ammonia. Terrestrial animals can't do that, right? Ammonia is not going to just seep out of our skin because air is not, ammonia is not soluble in air. So these guys, they use the kidneys, and some of them use it more than others for this reason, but it's not quite the same as it is for terrestrial organisms because they are living in an environment where they can quickly get rid of ammonia right from the start. They don't have to turn it into something less toxic. Okay, but they all have kidneys and they all work. And one of the other main functions is to regulate the ions in the bloodstream, right? And so we'll talk about that later. But the main idea is that we have to keep the right concentration of water compared to other stuff, right? So most of us are going to be, I'm just going to make up a relatively high proportion of H2O, right? And then if that's our general concentration of H2O in our cells, in our plasma, in our tissue, then everything else will be 15% solutes, stuff that's dissolved in water. And we have to maintain that way. But if you're in different environments, then that might be disrupted. And so you have to have some mechanism to keep it that way. And that's called homeostasis, right? Not just water and ions, but equilibrium, basically, right? And the main reason why this is true, for a variety of things, is that if you change that, then you change the environment that all of our things are existing in. Inside, if you look inside the cell, and you change the environment inside the cell, then that's going to have a big impact on enzymes. Because if you change that, what happens to enzymes? That are proteins, by the way. Most of them are. What do you do to a protein if you change its environment? You change its shape, right? And if you change its shape, then you're automatically going to change how it works. And so we have to keep everything the same. Otherwise, there are other things that happen, but this is the main thing. These are controlling all the chemical reactions that have to take place. If you change those, it's not going to work right. And so fishes use the kidneys for this, which could also affect enzymes, right? If it builds up, I mean, that's what kills us, basically, right? That's what's toxic. We're changing these things and they don't work properly, and some important metabolic process doesn't happen and the cell dies. And then, of course, the organism dies as a consequence. So we're changing that. We're getting rid of these things that are toxic, but we're also making sure that the relationship between water and everything else stays the same using the kidneys. And that plays a role in what the kidneys look like in different types of organisms. Okay, so as far as how the kidneys work, the functional unit of the kidney is called a nephron. And the nephron consists of different components, and so it's kind of hard to see on this thing, but you guys have it. So this little cup-shaped structure is part of the nephron. That's the Bowman's capsule. And then you've got the tubule, and so the main point here is that the tubules are passageways, but the other thing that you'll notice when you look at this slide is that the tubules also have another component that's closely associated with them, and that is part of the circulatory system, right? So these blood vessels are running and sometimes looping around the tubules and other ducts that make up the kidney to expose what's in that to the bloodstream, because that's what we're trying to do. We're trying to pass stuff into the bloodstream and then maybe pass stuff back into because all of this happens because membranes are permeable to some things, not other things. But you can't have membranes that are permeable to some things, not other things, all in the same spot, right? Because then you have just random movement of molecules again. So our kidneys and our nephrons are built by using that different permeability to accomplish what the kidney needs to do. So tubule is part of it. And then the collecting duct, again, which is essentially what is collecting the material that ultimately passes through the tubule that doesn't get resorbed back into the bloodstream. And then the other big thing is that it's not part of the nephron, it's part of the blood, the circulatory system. That is the glomerulus, right? And so that's this little ball of capillaries right here. Again, the point of the glomerulus is to take this relatively large blood vessel and turn it into a bunch of small ones, because now you want stuff to pass through the vessels of those capillaries into the Bowman's capsule because you're trying to get rid of stuff. Okay, so that's where it happens, and so that is the main thing that's different between different types of fishes. So in freshwater organisms, you have lots and lots and lots of glomeruli in the kidney, and they're large. What's the significance of that? Lots of what? As a consequence of all these big balls of capillaries and lots of them, what is the end result of that? A high, very high, super high, it's so high. What? Not metabolism, just the physical nature of this. Right? I've got a bunch of these really complicated networks of capillaries, a whole bunch of them, and they're large. So as a consequence of these little finger-like projections, as opposed to my arm, what happens as I take this and make it a bunch of small ones? What increases?
Blood flow.
Blood flow, not necessarily the rote, but surface area, right? The rate. I said rote, not necessarily the rote. Not necessarily the rate, but the surface area. So, and that's what we're relying on. We want to get stuff out of the bloodstream. The more space you have for that, the more efficient it's going to be. So if I have a bunch of them, then that's going to happen a lot. If I have a few of them that are small, then that's going to happen a lot less. But that's helpful for some fishes, right? And so the main difference, again, I'll talk about this again later, but the main difference we see here is that for marine organisms, those are animals that live in salt water. There's fewer of them, and they're smaller, which means they have overall a lower what? Surface area for transport of material. And so that's important because one of the things that happens as the blood enters the glomerulus in close proximity with this Bowman's capsule is water and metabolic byproducts, which would include ammonia, right? All the things that happen through the metabolism, move from the bloodstream into the into, not the tubule, into the Bowman's capsule, right? But ultimately into the tubule. So they're moving through the bloodstream into the tubule via the Bowman's capsule. So this might give you a hint, right, as to what's going on. If you have a lot of those, then you're ultimately going to collect a lot of blank in the tubules. What is it? If you're this animal and you have a lot of large glomeruli and this is happening, then you're going to get a lot of blank in the Bowman's capital. But, yeah, byproducts, but you skipped over one of the things. Water, right? So in other words, water and all that stuff is rushing out of the bloodstream into the Bowman's capsule, into the tubules. If you have a lot of them, then that's happening at a huge rate. If you have fewer of them, then that's happening more slowly. And that relates to the environment that these animals are living in, right? So if this is your concentration of water, or your relationship of water to solutes in the plasma, and you're living in an environment that is 95% H2O and only 5% solutes, then you have somewhat of a problem. And you have lots and lots of surface area exposed to water at the blank gills, then what's going to happen?
Water's gonna come into the body.
Osmosis is going to happen. Water is going to seep into the bloodstream, into the cells, and disrupt that. So what do you have to do?
Pump water back in.
You got to get it out. One way to do that is to have a whole bunch of large blanks. We're having an exam today. Filling in the blanks. I'm just going to write this stuff down and you can just do it. Yeah, glomeruli. If you've got a lot of glomeruli, you're getting a huge amount of water passing through that system and then just whoop, right out through the urogenital pore. If you're a marine organism, that's not the situation, right? If you're a marine organism, you're living in an environment that is 20% solutes, lots of salt in the marine environment. If there's 20% solutes, what's the proportion of water? 80. Now you have a problem still, but it's different, right? What's the problem now? You still have huge surface area exposed to this environment at the blank. Gills, there you go. There'll be like 4 questions on the exam. I hope you guys know what goes into blank. So you've got a lot of places where instead of now absorbing a bunch of water from the environment, what's happening?
You're losing them.
It's leaking out, right? It's leaking out. This water is leaking into this environment, and that's disrupting homeostasis, affecting the enzymes. So what are you going to do to stop it? Have fewer, smaller glomeruli. Yeah, you're going to lose some water through that process. We have to. Why do we have to do that?
Less surface area?
What's that?
Less surface area for it to lose it?
No, well, you're right. There's less surface area, so less water is getting out. But some is getting out, even though you don't want it to. But we still have to do that. Why?
It's got byproducts.
Got to get rid of the other stuff, right? We're going to produce ammonia. As we break down proteins, that has to happen. You got to get rid of it. There's no getting around it. But what you can do is get rid of it and limit the amount of water you're losing with it. And so that's why these guys have much smaller and fewer glomeruli, because they're trying to prevent water from seeping out through the membranes of the gills. And they also do some other stuff to help with that problem, and we'll talk about that later. Okay, so the idea here is that, of course, you've got the blood bringing all this stuff to the glomerulus and then the associated Bowman's capsule. It passes from the bloodstream into the nephron, and then, but some of the stuff that passes through that you might need, right? So as all this stuff is in the bloodstream, this Bowman's capsule has to let it in, just because of the permeability. In other words, to let the ammonia in, you also have to let the glucose, the amino acids, the sodium chloride in as well, right? So we're just dealing with these physical screens. If you make it so that ammonia can pass, then these other molecules can also pass. And so now what happens is this stuff gets into these tubules, which also have the blood vessels associated with them. The tubule has different permeability characteristics. So now what we can do is, okay, now we've got everybody in here. Some of you get back into the bloodstream. The rest of you guys out with you, right? And so that's why we have to do it in different places, because you have to rely on the permeability characteristics of the membranes that separate that. So some of this stuff, glucose, of course, which is what we use to produce ATP, amino acids, what we're using to build, proteins. Some of these ions that are important in muscular control, you still have to maintain homeostasis and all these other things. And so that stuff gets back into the bloodstream via the capillaries that are associated with the tubule. Okay, so all that together is a way that animals can do two things: get rid of nitrogenous waste and also maintain this homeostasis in terms of the relationship between water and solutes. Okay. Let's see. All right, so I have a few minutes. Okay, so now what I want to do is kind of focus back onto the reproductive system. So that's what the urinary system mostly does, and that now, as I said before, as part of that system, a lot of fishes also have elements of the reproductive system that are associated with it. And so we'll talk about the testes and their association with the kidneys first, and then we'll talk about the ovaries and some of the variability among the main types of fishes. So most fish are dioecious, which means there's two separate houses, right? There's one house for males, one house for females, and of course we call those males and females. And so males have testes and females have ovaries. It's typical that for most fishes the testes are paired structures. They're typically close to, like morphologically in proximity to the kidneys. As I said, some of them are elements of it that connect it to each other. So they're usually, when you dissect a fish, you'll see the ovaries or the testes, and then you also see the kidneys closely associated with that. The main thing that I'll kind of describe to you today and on Thursday is the degree of the association. So some have quite a bit of association, some have less, and that's kind of what I'm going to focus on. So for chondrichthyes, there's a strong connection between the two elements, and so much so that their kidney is described in different ways, right? And so the urinary kidney is part of the kidney that is responsible for, of course, filtering out the nitrogenous waste or ammonia, or in this case urea, and then depositing it into the duct that transports it out, and that's called the mesonephric duct. And then the sexual kidney is the part of the kidney that transports sperm into the tube that carries sperm cells, and that's called the vas deferens. For these guys, all that gets ultimately transported to the cloaca, and then ultimately is released from the organism. But of course, the thing that makes it the cloaca is that the other thing that empties into it is the digestive system. So it's a common chamber that receives all the material that's associated with that. The difference comes in the morphological part of it, and so for that I'm just gonna give you some drawings so you can see, and they'll be rough drawings, but I think you'll be able to see what's happening. Okay, so the main thing here is for the chondrichthyan fishes, you have two parts of the kidney. You got the urinary kidney, and then the sexual kidney. And so I'm just going to kind of do a rough diagram. So here is the kidney, and here is one of the testes. And then this is anterior, which means what?
Head.
Well, front. And posterior is the back end of the organism.
Is that how you spell posterior? Posterior.
Yeah. Okay, so and front, back, kidney, testes. Okay, so maybe just a little bit closer. So in chondrichthyan fish, the testes and the kidneys are very closely associated with each other. And so much so that the material that is released from the kidney, from the testes, passes through the kidney. And so you've got something like this. Right, so in other words, in the testes, you've got sperm being produced, and that passes through these canals that are part of the kidney. And that's why this is called the sexual kidney, because it's not performing that other function of removing nitrogenous waste. It is only transporting sperm cells from the testes into this tube, which is the vas deferens. Does that make sense so far? So here is the sexual kidney, and this is the urinary kidney. And that's where nitrogenous waste removal is happening for the most part. So that's where the functional nephrons are in chondrichthyan fishes. And then that material also, of course, has to be removed from the organism. Like that, right? So this is all nitrogenous waste going into this tube. And this tube that transports the nitrogenous waste is mesonephric duct. But you can see in chondrichthyan fish, that's the same. Which is the point, right? So in these guys, the testes and kidneys are closely associated, so much so that the kidney is only passing through sperm cells into this duct. The term that we use for the ducts that transport sperm is vas deferens. But in this case, vas deferens and mesonephric duct is the same, continuous with it, and then ultimately this empties into the cloaca, and then the cloaca will be emptied into the environment. Okay, I'm gonna stop. It's time to go. When we come back, we'll talk about